Welcome to the Step 1 Review Study Guide, and in this lesson, we learn about DNA replication. DNA replication is similar in eukaryotes as it is in single-celled prokaryotes. However, eukaryotic DNA replication is more complex. Both use many of the analogous enzymes. As well, DNA replication is semi-conservative. DNA has two strands, each running antiparallel to each other. DNA can only be synthesized in the 5' to 3' direction, and therefore involves continuous and discontinuous synthesis. The strand that is discontinuous is called the lagging strand. DNA replication starts at a particular sequence of DNA in an area rich in thymine and adenine base pairs known as the Tata box. This sequence is the origin of replication, and for prokaryotes, there is just one single origin of replication, but for eukaryotes, there can be many. The DNA must be pulled apart for the replication machinery to do its work. An enzyme known as helicase is responsible for unwinding DNA. And this Y-shaped region of DNA being pulled apart is known as the replication fork. Remember that helicase halves the DNA. In a condition known as Bloom syndrome, helicase is deficient because of the mutation in the BLM gene. When the DNA is uncoiled, it is reactive and not stable in this conformation. In order to prevent the DNA from reannealing, proteins called single-stranded binding proteins bind to the single-stranded DNA. As the helicase is being unwound, this creates supercoils, which, like a pair of supercoiled headphones, makes it even more difficult to unwind. The enzyme topoisomerase creates single or double-stranded breaks in the helix that adds or removes supercoils. Drugs have been developed to inhibit topoisomerase. In eukaryotes, irinotecan and topotecan inhibit topoisomerase 1, and etoposide and teniposide inhibit topoisomerase 2. In prokaryotes, fluoroquinolones inhibit topoisomerase 2, also known as DNA gyrase, as well as topoisomerase 4. DNA can only be synthesized on an existing ribose 3' hydroxyl. The enzyme primase makes an RNA primer to which DNA polymerase 3 can start replication from. DNA polymerase 3 is a prokaryotic polymerase that elongates the leading strand by adding deoxynucleotides on the 3' end. In the lagging strand, it will keep adding nucleotides until it reaches another RNA primer. This creates what are known as Okazaki fragments. DNA polymerase 3 synthesizes in the 5' to 3' direction but proofreads using 3' to 5' exonuclease. Some drugs can block DNA replication by having a modified 3' hydroxyl, which prevents elongation, terminating the chain. DNA polymerase 1 is only in prokaryotes. This enzyme degrades the RNA primer in the 5' to 3' direction, the enzyme exonuclease. Then it replaces the primer with DNA. DNA ligase 1 joins DNA fragments in the lagging strand by the formation of a phosphodiester bond. Remember that ligase links DNA. The final enzyme is telomerase. Telomerase is an enzyme only in eukaryotes, and it's a reverse transcriptase which elongates the telomeres by adding the sequence TTAGGG to the 3' ends of chromosomes. This is because the ends of DNA become shortened after each new replication. By adding to and extending telomeres, this can avoid the loss of genetic material. Often, if the function of telomerase goes unregulated, it could cause cancer, which allows unlimited replication. And here's a video of the whole machinery working in action in a highly accurate computer simulation. That's all for this lesson of Step 1 Review. We'll see you in the next video.